Over these microRNA, can you describe what they do briefly for a lay audience?
Yes. So actually, this is a Nobel Prize winning about 7 years ago. So it was discovered that we have actually, so normally how it goes, you have DNA, RNA, proteins, right?
Right.
So the DNA codes for RNA, and the RNA code translate to codes for RNA, and the RNA translate to protein. But it was discovered that we have actually small RNA molecules, which are therefore called microRNA, and they are not coding for any proteins, but actually they have regulatory function. So they bind genes, and it's estimated that around 60% of all of our genes are regulated by these microRNAs. We have about 700 of them.
Are these the generals in the army?
These are kind of, I don't know, because these are maybe the most sophisticated means to control the genes, because one microRNA can control the function of around 300 genes.
Wow.
Because they have special requirements, but they are quite broad, so they can bind to many different genes.
So they're like the killer cells in another sense that have this multifunction.
Right, exactly, right. So actually they bind in the, so you have a gene, or you have an RNA, and there's a long pieces of this RNA which are called the 3' untranslated regions. So they do not code for anything. And people will wonder for years why they are there. So you have a big chunk of RNA which is code for nothing. So why do you need it? And it was discovered that these microRNAs actually bind in the 3' UTR of the gene, and that's how they regulate gene expression.
I see. So
what's interesting is that in 2004, it was discovered that viruses, not HIV, but other viruses, contains microRNAs. Completely amazing. So the viruses themselves, especially from the herpes family, for example, CMV, EBV, a virus that can cause cancer, KSHV also can cause cancer, and each one of them encodes its own microRNA. And the question is what they are doing. And what we have shown is that when the virus infects the cells, some of these microRNAs simply target the proteins which will be recognized by the natural killer cells. So it's fascinating. So I just told you previously that NK cells should kill the infected cells if they recognize something on the infected cells. What these microRNAs, so when the virus infects the cell upregulates something what we call stress-induced protein. So the cell feels that it's under stress. It's upregulates the expression of this stress-induced protein. And NK cells have a special killer receptor that will recognize this stress-induced protein. And what these sophisticated viruses are doing, they simply design their own microRNA to hit the same protein and to prevent its expression. So now the NK cells can no longer kill these targets.
So how do you get it then that you have this kind of war in which everybody's adjusting to the other side?
Right.
I assume the viruses adjust back again.
Right.
And so what do they do to protect them from getting killed?
It's exactly like that. So there is always a balance between the viruses and the NK cells. And actually, viruses, they are so sophisticated. They develop such a fantastic ways to manipulate the cell, and we learn so much from these viruses because. So now you find, for example, a microRNA of the virus that target this gene. Now we can discover cellular mechanisms which uses similar microRNAs which belongs to the host and target the same genes under certain conditions. So this is exactly that. This is a war between the virus and the immune system, and there is always a balance between these two systems.
And how does that help you in dealing with AIDS in a very practical way?
So now this is one of the current work that we are doing with a group of Frank Plummer and Adrian Myers.
And Plummer is in Winnipeg.
Exactly.
That's the big
exactly. So one of he is very famous and very well known for one of the maybe best discoveries in AIDS research. He discovered that in Kenya, there are women which are definitely were exposed to HIV, but still they are not infected. And no one knows why. How come these women do not get infected? And if we will be able to maybe discover the reason for that, you might be able to give a better solution to HIV-infected people. And one of the ways that we are thinking of going is by testing the microRNAs in this group of women. So we will take the infected cells from women that are infected with HIV, with women from healthy women, and from these resistant women in Kenya.
So you have 3 groups.
Exactly, and we'll try to see what is the microRNA phenotype of each woman, and then maybe from that to learn of the ways by which maybe the resistant women are resistant to HIV.
How do you start? Do you do the analysis of the makeup of it, or do you do actual experiments in a dish of what happens?
So actually first people from, some students from my lab went to Kenya and worked with the Frank's group, especially with Adrian. And we just collected T cells or immune cells from these 3 groups of women. It took about 2 months, I think. And then we came back to Israel, and now we are going to establish this research. We are going to do this to start with just maybe a profile of all of these microRNAs, and then to keep going. And there are hints in the literature. It's not just a random research that you look for something. There are hints in the literature there is something going on with these microRNA in the infected patients.
It's very, the difference between these big random studies versus these very targeted studies. I find the targeted studies very interesting than the big random ones.
But sometimes you need scientific lead. So how can you go, so you have a group of women which is resistant. How can you go about researching them? Sometimes you need to start with a scientific hint, especially if you have some scientific background to say that there is something in the microRNA there. So you have to start with some broad.
So what's your hypothesis?
I think that by the way, we start the same with the NK cells and pregnancy. We also started from a broad analysis of the genes which are expressed in these NK cells from these women. I think that we might be able to find in the resistant women certain classes of microRNAs that are unique to these women. I think so.
That's your hypothesis.
Yeah, I think so.
And how long do you think it'll take? You don't know when you go into these.
I mean, I don't think it will take too long. No, because now everything is set up. I mean,
the most. Did you get a big grant for this? Oh, you got a big grant from Gates, didn't you?
No, not me.
No,
Dr. Plummer got.
Plummer,
we got actually a grant from from Manitoba.
Yeah.
To do this type of research. Actually, it's not. I don't think it's. I mean. But still, it's okay. So
you need more money.
Yeah, like
you're shy. But
yeah, I mean, in Israel, the money is always a problem.
Well, it's in all labs, it's just worse here.
It's much worse.
But partly because you have so many good scientists makes it worse.
Right.
So the competition is greater
right.
for a lesser pot. No, it makes it tough because you can't do your work without the money.
Right.
And the work is critical.
So it was really helpful. So here it was not only the money. We also the fact that Plummer Group identified these women, and we have access to this women. This just enabled the research. I think it's going to be really exciting.
So you're really expecting.
Yeah, I am.
And how many people in your lab?
I have around 15 people.
15? You're super.
Yeah.
How many postdocs?
I don't have any postdocs. They're all PhD students.
You don't have any postdocs?
No, actually, that's almost all labs in Israel have mainly PhD students.
But that's a weekness, isn't it?
It is. On one hand, it's a weakness. On the other hand, is a great advantage because these PhD students are really fresh and naive, and sometimes you need to have a naive attitude and a fresh attitude to do a good research. So actually, I'm very proud. I have many students that I taught, and now they went to the US or to Canada to do their posts, and they will come back to Israel as a independent scientist. So I really like it.
I see. Well, with that, we'll end, and I want to thank you, and thank you not only for this brilliant research, but you've somehow retained a kind of naive attitude toward that you say about your students, this kind of eagerness and keenness and openness, I think, that makes it great work. Thank you.
Thank you.
Will Howard Cedar win a Nobel Prize? Next week's guest showed how methylation made DNA open and sensitive rather than closed and inaccessible. This pioneer in epigenetics explores how genes are turned on and off. Does biology provide insights into society? When do Jews get turned on or off religion? How do Jewish youth get turned on to defend Israel? Join us next week for Switching.